Welcome to this deep dive into the RadOnc Smart Review RadBio edition. We are guides today,
Hello, everyone.
and today we're really going to get into two concepts that are, well, absolutely fundamental in radiation biology, no matter where you are in residency.
That's right. We're talking linear energy transfer, LET, and relative biological effectiveness, RBE.
Exactly. And understanding these is just crucial. It goes beyond just knowing the dose in gray.
It really does. It's about the quality of the radiation, how it actually deposits energy and, causes biological effects. It helps understand why some radiations are different.
So it's not just the quantity, but the quality of that punch, like you said.
Precisely.
Okay, so let's start at the beginning then. Linear energy transfer. What exactly is LET?
All right, LET. So at its core, LET measures how much energy a charged particle deposits as it zips through tissue or any medium, really.
Like a track of energy.
Exactly. It's the average energy deposited per unit length along that particle's track.
Okay, and units. This sounds like something important for exams.
Oh, definitely. If you remember one thing about LET units, make it kilo electron volts per micrometer.
Kilo electron volts per micrometer. Got it. So kevim.
Yep, lock that one in. That's the standard.
So we often hear about low LET versus high LET radiation. Can you break that down for us, maybe with some examples?
Sure. So low LET radiation. Think of your standard photons like X-rays and gamma rays and also electrons.
Or daily workhorses in the clinic.
Right. We call these sparsely ionizing. Imagine maybe like a ping pong ball bouncing sort of randomly through a room.
Okay.
It hits things here and there, transfers a little energy with each collision, but the interactions are pretty spread out along its path. It's not a very dense track of hits.
That makes sense. Scattered interactions.
Yeah.
So what about high LET then?
High LET is the opposite. Think alpha particles or neutrons.
Wait, neutrons aren't charged, right? How do they fit in?
Good point. Neutrons themselves are neutral, but they interact with nuclei in the tissue, mainly knocking protons forward. These recoil protons are charged and deposit energy very densely.
Ah, okay. So it's an indirect effect causing dense ionization.
Exactly. So for high LET, instead of the ping pong ball, picture a bowling ball crashing through.
Right. Much more impact, much more concentrated damage along its path.
Precisely. It leaves a dense trail of ionization. That's high LET, densely ionizing radiation.
If we could visualize these tracks, say in a simulation.
Yeah, you'd see a clear difference. A low LET track, like from an electron, might look kind of sparse, wandering a bit, with little bursts of energy deposited here and there.
Okay.
But a high LET track, from an alpha particle perhaps, would look much straighter and incredibly dense, like a thick, solid line of energy deposition.
And that dense deposition, that's key for biological damage.
Absolutely. That concentration of energy makes it much more likely to cause complex, harder to repair damage, especially to critical targets like DNA.
Now, you mentioned LET as an average. Does that mean it can vary for a single type of radiation?
Yes, that's an important point. Often a beam isn't monoenergetic, so we talk about average LET. And there are actually two main ways to calculate this average.
Okay.
First is the track average LET. You basically take the total energy deposited along the whole track and divide by the total track length. Simple average over distance.
Right. Equal weight to each part of the track.
Exactly. The second way is the energy average LET. This one's a bit different. You look at segments of equal energy deposition, say 1 kilo electron volt, and average the track lengths needed for that deposition.
Okay. So it weights things differently.
It does. It tends to give more weight to the parts of the track where energy deposition is denser, where the LET is higher.
And is one more relevant than the other? I think I heard something about neutrons.
Yes, especially for neutrons, the energy average LET is generally considered more biologically relevant. It better reflects the impact of those densely ionizing recoil protons.
And the values could be quite different.
Oh, very different. Take 14 mega electron volt neutrons, for example. The track average LET might be around, say, 12 kilo electron volts per micrometer.
Okay.
But the energy average LET for those same neutrons could be closer to 100 kilo electron volts per micrometer.
Wow, that's a huge difference, nearly tenfold.
It is. And it highlights why that energy average matters more for predicting biological effects with neutrons.
Good to know. Okay, could you give us a feel for the range? Some typical LET values for radiation types we might encounter.
Sure. Let's start low. Cobalt 60 gamma rays, very low LET, maybe 0.2 kilo electron volts per micrometer.
Okay, very sparse.
Then maybe 250 kilovolt peak X-rays, the old ortho voltage standard, around 2.0 kilo electron volts per micrometer. Still quite low.
What about protons? We use those clinically.
Right, so high-energy protons, like 150 mega electron volts used in therapy, actually have a low LET in the entrance region, maybe 0.5 kilo electron volts per micrometer.
Lower than the KV X-rays even.
Initially, yes, but if you look at lower-energy protons, say 10 mega electron volts, the LET goes up, maybe 4.7 kilo electron volts per micrometer. Low to intermediate range there.
Interesting. And then the high LET ones?
Well, we mentioned fast neutrons, like those 14 mega electron volt ones, with an energy average LET around 100 kilo electron volts per micrometer. That's definitely high LET territory.
Okay.
Alpha particles, say around 2.5 mega electron volts, are even higher, maybe 166 kilo electron volts per micrometer. Very high LET.
And then things like heavy ions.
Oh yeah, they can go extremely high. A heavy ion like iron, accelerated to 2 giga electron volts, could have an LET around 1,000 kilo electron volts per micrometer. Just incredibly dense ionization.
That really paints a picture of the huge spectrum we're dealing with. Is there a general rule, like how a particle's energy affects its LET?
Yes. Generally, for a specific type of charged particle, there's an inverse relationship between its energy and its LET.
Inverse. So higher energy means lower LET. That seems counterintuitive at first.
It does, but think about it. A faster particle, higher energy, spends less time near any given atom as it flies past.
Less dwell time, fewer interactions per unit distance.
Exactly. So lower LET. As the particle slows down, it interacts more strongly, and its LET increases.
Which is the basis for the Bragg peak in protons, right? They slow down at the end of the range?
Precisely. The LET goes up significantly right at the end of the track.
Okay, I think we've got a good handle on LET, the physical side. Now, let's connect it to the biology with relative biological effectiveness, RBE.
Right, so RBE bridges the gap. LET is the physics, RBE is the biological consequence. It tells us how effective different types of radiation are at causing a specific biological effect compared to a standard.
So it's like a conversion factor.
Yeah.
From physical dose to biological impact.
Sort of, yeah. It's a relative measure. The formal definition is the ratio of the dose of a standard reference radiation needed for a specific effect, divided by the dose of the test radiation needed to produce the exact same level of that same effect.
Right, so the formula is RBE equals dose of standard divided by dose of test for the same endpoint.
Exactly. Keep that formula in mind. Dose standard over dose test.
And what's the usual standard radiation we compare against?
Historically, and often still, it's 250 kilovolt peak X-rays.
The orthovoltage X-rays again.
Yep. Sometimes you might see cobalt-60 gamma rays or other photon energies used as reference now, but 250 kVp is the classic.
And by definition, the RBE of that standard radiation is?
Always 1. It's the baseline you're comparing everything else to.
Okay, can we walk through an example? Make it concrete?
Let's do that. Imagine we're looking at lethality in mice specifically. The dose needed to cause 50% mortality in 30 days, the LD5030.
A common radiobiology endpoint.
Right. So say we find experimentally that it takes 6 gray of our standard 250 kVp X-rays to achieve LD5030 in a group of mice.
6 gray of X-rays.
Then we do the same experiment with fast neutrons, and we find it only takes 4 grays of neutrons to get the same 50% mortality.
4 gray of neutrons. Less dose for the same effect.
Exactly. So to calculate the RBE of the neutrons for this endpoint,
We use a formula. Dose of standard divided by dose of test. So 6 gray divided by 4 gray.
yep, which equals 1.5.
So the RBE is 1.5. What does that tell us?
It tells us that for causing this specific LD5030 endpoint in mice, these fast neutrons are 1.5 times more biologically effective per gray than the standard X-rays.
Got it. You need less neutron dose for the same biological damage.
Precisely.
Are there any, major caveats we absolutely need to remember about RBE values?
Oh, definitely. 3 huge things. First, RBE is relative. It only has meaning in comparison to a specific standard radiation.
Okay, always need the reference.
Second, RBE is experimental. You can't just calculate it from first principles. It has to be measured in biological experiments.
Makes sense. Biology is complex.
And third, RBE is specific. The value depends critically on the biological system, the cell type, the tissue, and the specific biological endpoint you're measuring. Cell-killing RBE might be different from mutation RBE, for instance.
So context is everything for RBE.
Absolutely crucial.
Okay, so we have LET, physics,
Hmm?
and RBE, biology. How do they actually relate to each other? Is there a predictable connection?
Yes, there's a very important relationship. Generally speaking, for very low LET radiation, our standard photons and electrons, the RBE is also low, usually close to 1.
Okay, sparse ionization, less effective per gray.
Right. The damage is often simpler, more easily repaired by the cell. Sublethal damage is more common.
And as LET starts to increase,
As LET goes up, the ionization events get closer together along the track. This denser energy deposition is more likely to cause complex clustered damage to DNA. Think of multiple lesions close together, especially double-strand breaks.
which are harder for the cell to repair.
Much harder. So as LET increases, the radiation generally becomes more biologically effective per unit dose, meaning the RBE tends to go up.
Does it just keep increasing indefinitely? Higher LET always means higher RBE.
Ah, no. And this is really fascinating. The RBE increases with LET up to a certain point, and then it actually peaks.
It's a peak RBE. Where does that happen?
The peak RBE, especially for things like mammalian cell killing, typically occurs when the LET is around 100 kilo electron volts per micrometer.
100 keV again. That number seems important. We saw it with the neutron energy average, LET.
It is a key value in radiobiology. It seems to be sort of the sweet spot for maximizing biological damage efficiency, particularly cell killing.
So what happens if the LET gets even higher, past that 100 keV peak?
Then we see something called the overkill effect. If the LET becomes too high, say well above 100 or 200 kilo electron volts per micrometer, the RBE actually starts to decrease.
Decrease. Why would it become less effective if it's depositing even more energy?
Because the energy deposition becomes so dense that you're essentially depositing more energy in a very small volume, like a segment of a cell nucleus, than is actually needed to kill that cell.
Like using a cannon to swat a fly. You mentioned that analogy earlier.
Exactly. You're wasting energy. That extra super dense deposition doesn't lead to proportionally more cell killing. Some of the energy is redundant for that specific endpoint, hence overkill.
That makes intuitive sense. Is there a deeper reason why the peak is right around that 100 keV value? Does it relate to cell structures?
The leading hypothesis ties it directly to the dimensions of DNA. The DNA double helix is about 2 nanometers wide.
Okay.
Radiation with an LET around 100 kilo electron volts per micrometer has an average spacing between ionization events that's, well, roughly on the same scale as the DNA diameter.
So a single particle track at that LET is optimally spaced to cause a double-strand break, hitting both strands efficiently.
That's the idea. A single track has a high probability of causing complex, lethal damage, like a double-strand break. At lower LETs, you might need multiple tracks or events close together. At much higher LETs, you get the overkill.
Fascinating. So if we
Right.
plotted RBE versus LET on a graph, what would that look like?
You typically plot RBE on the y-axis and LET on the x-axis, usually using a logarithmic scale for LET because the range is so wide.
Right.
And the curve would start low at low LET, rise up to a distinct peak around that 100 kilo electron volts per micrometer mark, and then gradually fall off again at very high LET values due to overkill. It's characteristic hump shape.
Okay, that visual helps solidify the concept. Now, LET is clearly a major factor driving RBE, but you mentioned RDE is complex. Are there other things that influence it?
Oh, absolutely. LET sets the stage, but other factors modulate the RBE significantly. One big one is the actual radiation dose level.
How does dose affect RBE?
Generally, for high LET radiation compared to low LET, the RBE is higher at lower doses and tends to decrease as the dose gets higher.
Higher RBE at lower doses. Why is that?
It relates to the shapes of the cell survival curves. Low LET curves usually have a shoulder region at low doses, meaning cells can repair some of the damage. Survival doesn't drop off purely exponentially at first.
Okay, repair capacity matters.
Right. High LET curves, however, tend to be steeper and more linear, almost from the start. The damage is less repairable, so killing is more efficient even at low doses. The difference in effectiveness, RBE, is therefore greatest at those low doses where repair is significant for low LET radiation, but not for high LET.
That makes sense. What about how we deliver the dose, like fractionation?
Yes, the number of fractions or the dose rate is also key. Fractionating the dose, or delivering it slowly at a low dose rate, allows time for repair between fractions or during the protracted exposure.
And repair works better for low LET damage.
Exactly. Cells repair low LET-induced sublethal damage much more effectively. High LET damage, being more complex, shows much less repair and therefore less dose sparing with fractionation or low dose rates.
So the RBE of high LET radiation would appear higher when using fractionation compared to a single dose.
Precisely. The relative advantage of high LET becomes more pronounced when you give the low LET radiation a chance to be repaired.
Okay. And the third factor you hinted at earlier, the biological system itself.
Absolutely. The biological system, or endpoint, is critical. Different cell lines have different inherent radiosensitivities and repair capacities. Different tissues behave differently.
Right.
And the specific biological effect you measure matters too. RBE for tumor control might differ from RBE for, say, late normal tissue effects like fibrosis or cataract formation.
So a tissue that's very good at repair might show a higher RBE for high LET radiation because the low LET radiation effect is reduced by all that repair.
That's generally the thinking, yes. The differential effect between high and low LET becomes larger in systems with significant repair capacity.
Okay, this is really adding layers to just thinking about dose. Let's distill this into some clinical pearls. What are the absolute must-know takeaways for residents?
Okay, pearl number 1. High LET radiation generally causes more complex DNA damage. This means it's usually more biologically effective per gray, leading to a higher RBE. This difference is often most pronounced at lower doses.
Got it. Higher LET, generally higher RBE, especially at low dose. Pearl 2.
Pearl number 2. RBE is not a fixed number. Remember, it's relative, experimental, and specific. It totally depends on the comparison radiation, the dose, the fractionation, the cell or tissue type, and the biological endpoint you care about. Context is king.
Cannot stress that enough. RBE needs context.
Yeah.
And Pearl 3.
Pearl 3. Understanding that RBE versus LET curve the rise, the peak around 100 kVim, the fall off due to overkill, is fundamental. It helps you interpret radiobiology studies and think critically about why we might choose different radiation types like protons or heavy ions for certain clinical situations.
Perfect transition.
Nah.
Let's talk about that clinical decision-making.
Yeah.
How do LET and RBE actually factor into choosing between, say, protons, neutrons, or heavy ions?
Right, this is where it hits the clinic. Let's start with protons. Protons, for most of their path into the patient in that entrance region, have a fairly low LET, similar to photons.
So RBE is low there.
Yeah, generally considered about 1.1, so maybe slightly more effective than photons, but not dramatically so based on RBE alone in the entrance.
But the magic happens at the Bragg peak.
Exactly. As the protons slow down at the end of their range, in the Bragg peak, their LET increases significantly, and so does their RBE.
But the main selling point for protons is usually
It's still primarily about the physics, that incredible physical dose distribution, the sharp peak, and the near zero dose beyond the target. That physical advantage is huge for sparing normal tissues. The RBE increase at the peak is sort of an added factor, but still subject to some uncertainty and ongoing research.
Okay. What about neutrons? It said they are high LET.
They are. Neutrons deliver high LET dose throughout the irradiated volume. This means a higher RBE pretty much everywhere compared to photons.
Any advantages to that?
Potentially, yes. High LET radiation also tends to have a lower oxygen enhancement ratio, the OER.
Meaning it's better at killing hypoxic cells.
Correct. Since hypoxic cells are often resistant to low LET radiation, the high LET and low OER of neutrons could be advantageous for certain aggressive, hypoxic, or traditionally radio-resistant tumors.
But neutrons have downsides too, right?
Definitely. Neutron beams are generally less precise physically than photon or proton beams, harder to shape the dose. And the RBE itself can be quite variable and uncertain in different normal tissues, leading to concerns about managing toxicity. Neutron therapy isn't widely used now because of these challenges.
Makes sense. Okay. Finally, heavy ions, like carbon ion therapy. What's the LET and RBE story there?
Heavy ions like carbon are really interesting because they combine advantages. Like protons, they have a Bragg peak, offering excellent physical dose localization.
Okay.
But crucially, in that Bragg peak region, or the spread out Bragg peak, the SOBP that we use to cover the tumor volume, the LET of carbon ions becomes very high.
So you get high LET right where you want it and the tumor.
Exactly. This results in a high RBE and a low OER, specifically within the target volume. So you potentially get both the physical precision and enhanced biological effectiveness for killing resistant or hypoxic tumor cells.
The best of both worlds.
Potentially, yes, for certain tumor types. But it's complex. Modeling the RBE for heavy ions accurately is challenging, as it changes rapidly with depth and depends on the tissue. There's also the issue of nuclear fragmentation. The heavy ions can break up, creating lighter particles that travel beyond the Bragg peak, giving a low-dose tail.
So still complexities to manage. It really shows how LET and RBE aren't just academic concepts, they directly impact the pros and cons of these advanced therapies.
Absolutely. You can't fully appreciate why we use particle therapy or its potential benefits and challenges without understanding LET and RBE. Just knowing the dose in gray isn't enough.
Couldn't agree more. Okay, I think it's time to test ourselves. Ready for the board blitz.
Let's do it. Hit me.
Question 1. Linear energy transfer, or LET, is typically expressed in which units? A gray, B sievert, C joules per kilogram, D kilo electron volts per micrometer.
That has to be d kilo electron volts per micrometer. We hammered that one home. It's energy per unit distance at the micro level.
Correct. Okay, question 2. Relative biological effectiveness, or RBE, is calculated as A dose test, dose standard, B LET test, LET standard, C dose standard, dose test for equal effect, D dose rate standard, dose rate test.
RBE compares test to standard, so you want to know how much less test dose you need. It's dose of standard divided by dose of test, so C.
Correct again. Dose of standard divided by dose of test radiation for equal effect. Question 3. For mammalian cell killing, RBE generally reaches its maximum value at an LET of approximately A1 keVim, B10 keVim, C 100 keVim, D 1000 keVim.
That peak, that optimal LET we discussed, linked to DNA dimensions, that's around C 100 kilo electron volts per micrometer.
Spot on. C is the answer. Last one, question 4. Comparing 1 gray of fast neutrons, high LET, to 1 gray of 6 mega electron volt X-rays, low LET, the fast neutrons would generally be expected to have A. Lower LET, lower RBE, B. Lower LET, higher RBE, C. Higher LET, lower RBE, higher LET, higher RBE.
Okay, neutrons are high LET compared to X-rays, and high LET generally means higher biological effectiveness per gray, so higher RBE. That means D, higher LET and higher RBE.
Perfect. D is correct. Excellent run through the blitz.
Good questions covering the key concepts.
So let's quickly recap the absolute essentials from this deep dive.
We defined LET, linear energy transfer, average energy deposited per unit track length. Units are key, kilo electron volts per micrometer. Differentiating low LET, sparse like X-rays, from high LET, dense like alphas or neutrons.
Right. Then RBE, relative biological effectiveness. It's that ratio. Dose of standard radiation over dose of test radiation for the same biological effect. Remember, it's relative, experimental, and specific to the system and endpoint.
And the crucial link. RBE generally increases with LET, peaking around 100 kilo electron volts per micrometer, then decreases due to the overkill effect. That peak relates to efficient DNA damage.
We also noted other factors matter for RBE, the dose level, fractionation or dose rate, and the specific biological system involved.
And bottom line, high LET radiation typically packs a bigger biological punch per gray, meaning a higher RBE, which is clinically relevant when comparing treatments like protons, neutrons, and heavy ions.
Absolutely. Grasping LET and RBE really elevates our understanding beyond just dose, helping us understand how radiation works and why we choose specific treatment approaches.
Couldn't have said it better myself. Foundational stuff.
Definitely. Well, that wraps up our deep dive for today. For more practice and review, you know where to go. RadonSmartLearn.com.
And make sure to subscribe to Rad1 Unc Smart Review so you don't miss our next exploration.
Thanks so much for tuning in.